In the spring of 2020, as the world locked down and hospitals filled, two small biotech companies, Moderna and BioNTech, were working on something that had never been done before. A vaccine built not from pieces of virus, not from weakened virus, but from a molecule of messenger RNA. Within a year, those vaccines had been administered to billions of people. It was the fastest vaccine development in human history. How did they do it? And how does an RNA molecule teach your immune system to fight a virus it's never seen? In Series 1, we covered the central dogma. DNA is transcribed into mRNA, and mRNA is translated into protein. Every protein your body makes, from hemoglobin to insulin to the antibodies that fight infections, is built by ribosomes reading mRNA. The key insight behind mRNA vaccines is elegant. Instead of injecting a protein or a dead virus to train the immune system, what if you inject the mRNA instructions? Traditional vaccines are like showing your immune system a wanted poster. mRNA vaccines give your cells a temporary script to perform a short scene. The specific target of the COVID-19 mRNA vaccines is the SARS-CoV-2 spike protein, the crown-shaped projection on the virus surface. This is what the virus uses to enter human cells by binding the ACE2 receptor. Blocking this blocks viral entry. But natural mRNA is notoriously fragile. Enzymes called RNases are everywhere and degrade RNA rapidly. To solve this, scientists replaced uridine with pseudoridine, a subtle modification that makes the mRNA less recognizable to immune surveillance. This modification was pioneered by Dr. Katalin Karikó and Dr. Drew Weissman, work that earned the 2023 Nobel Prize. They also optimized the 5U cap and the poly-A tail to maximize stability and translation efficiency inside the cell. Then came codon optimization, selecting the specific triplet codons that human ribosomes read most efficiently. Scientists also introduced the 2P modification, two proline substitutions that lock the spike protein in its prefusion shape. But RNA cannot enter cells alone. The solution? Lipid nanoparticles, or LNPs. Think of them like a soap bubble protecting the mRNA. An LNP is a tiny sphere made of 4 lipid components, designed to evade detection and fuse with cell membranes. The key is the ionizable lipid. It's positively charged to bind mRNA during manufacturing, then becomes neutral to reduce toxicity. Inside the cell's acidic compartments, it becomes charged again, destabilizing the membrane and releasing the mRNA. This technology was developed largely by Peter Cullis and his team, building on decades of basic research in lipid chemistry. After injection, LNPs are taken up by muscle cells and dendritic cells, the sentinels of the immune system. These cells read the mRNA, produce spike protein, and display fragments of it on their surface using the MHC presentation system. B cells recognize the spike and produce neutralizing antibodies that block the virus from engaging your cells. Meanwhile, T cells learn to coordinate the response and eliminate infected cells. Long-lived memory cells persist for years. The mRNA itself is completely degraded within days. It cannot enter the nucleus and leaves no permanent trace. The COVID-19 vaccines prove the platform. Now mRNA technology is being applied to a stunning range of conditions. Personalized cancer vaccines are in development, training the immune system to attack a specific patient's unique tumor mutations. It's also being used for rare genetic diseases, periodically restoring a missing protein like those needed for cystic fibrosis. This platform may be among the most medically transformative innovations of our century. Visit unbedsidemanners.com for more stories.